Click for IB Biology. This is the first video in a series concerning itself with the origin of cells. We're looking at Earth's early atmosphere during a period known as the Hadean and Archean periods, approximately 4.5 to 3.5 billion years ago. And the nature of the reducing atmosphere, the anaerobic conditions that were present, and the eventual evolution of the Cyanobacteria. There's a PDF of this presentation down here in the description, and there's a quiz for you to take also to check your understanding of this content. So we're looking at the formation of cells, but not in the nature that you will look at in the next section, the complex eukaryotic cells. Here we're way back looking at protocells. One of the fundamental things for there is the formation of a cell membrane. Let's take a look at the atmospheric conditions. Well, these were dominated by things like ammonia and carbon dioxide. It's believed that there were others around, such as nitrogen, some water, methane, hydrogen, and carbon monoxide. And the period of time we're discussing is about 4.5 billion years ago in the Hadean period, eon, actually. The energy sources available at this time were in the form of UV light, and also atmospheric lightning might be suggested. And the idea that we're looking at is that these simple molecules, inorganic molecules, would be combined using these energy forms in order to make the simpler biological molecules, such as proteins, carbohydrates, and lipids, and perhaps the nucleic acids. Experimental work carried out in the 1950s by Miller and Urey attempted to reconstruct the Hadean atmosphere in the laboratory within their glassware, in which they placed water, methane, ammonia, and hydrogen. They included in their simulation of the atmosphere a spark from the electrodes and some heat as well. They also had a cooling jacket, which meant that they could have cycles of hot and cold as the molecules began to emerge. So this would cycle round in circles driven by the heating process, and was allowed to carry on for a period of time. In their identification, Miller and Urey identified simple monomers such as amino acids and sugars, the details of which you can find in the biochemistry section of this video playlist, and indeed some early signs of polymers with amino acids forming things like dipeptides and disaccharides as well, so double sugars. So these are joining together, they're being made, but then they're also joining together to make the simpler polymers. Interestingly, although the work was carried out in 1952, at the completion of their work, the flasks were sealed and were actually reopened in 2008 and subject to 21st century chemical analytical tools. When this was done, it was discovered that these tools, this instrumentation was far more sensitive than that available to Miller and Urey, and a larger range of amino acids was found to be in their flask, and indeed sugars, and in fact longer polymers were also found there, some of which were early versions of the nucleic acids, such as the RNAs. the atmosphere, the Hadean atmosphere, which operated 4.5 billion years ago, all the way through to 3.5 billion years ago, was largely anaerobic and largely a reducing atmosphere. And these conditions actually are ideal for the beginning of the manufacturing of polymers. It's only towards the end of the Archean period that we began to see. By that time, we already had Cyanobacteria, and they would be carrying out simple forms of photosynthesis, producing an early oxygen atmosphere for Earth. So during this 4.5 to 3.5 billion year change, at some point these polymers started to form enclosed simple structures, simple vesicle style structures that were the early protocells that we'll discuss in other videos in this sequence. Those videos and links to those videos are appearing before you now, along with a subscription button to the channel, and the playlist is displayed so that you can look at the choice of videos for you to consider in the origins of cells.